nice introduction. I'll just see if I can share. Okay, can you see that PowerPoint presentation? Yep, great.
Yes.
All right. Thank you again for that nice presentation. So I'm looking forward to talking to you all today about some of the research that we do in my group here in Sydney in Australia. And today I'm going to talk to you about marine microorganisms and how their behavioural interactions can lead to some quite profound implications for a variety of processes that occur in the ocean. So the marine microbes I'm going to focus on are those that live in this environment, the pelagic ocean. So planktonic microbes floating around out in the open water. And these microbes are highly abundant and they're also extremely diverse, and they play a lot of really important roles in controlling the function of the ocean. Oops. And among these roles is their important activities within the ocean's biogeochemical cycles. So to portray that, what I'm showing here in this cartoon is the ocean's carbon, nitrogen, and sulfur cycles, which are, of course, key parts of the biogeochemical cycles within the ocean. And I know there's a lot going on in this image, so I'm not going to go into any of the specific detail, except to say that the main take-home message is that it's different groups of microbes, largely within the water column, that control the chemical transformations that make up these biogeochemical cycles. And this is important because this ultimately influences things like nutrient cycling, which controls the productivity of marine food webs, and it also mediates the fluxes of climatically important gases in and out of the ocean into the atmosphere. And this is how the ocean can have an influence on our global climate. Now, while all of these processes are important at global ocean scales, many of the chemical transformations that build these biogeochemical cycles actually occur at much smaller scales, really minute scales that, in fact, take up a fraction of an individual drop of seawater. So these occur in specific microenvironmental scenarios and ultimately play really important roles in the ocean. And I'm just going to give you a couple of examples of some of these. So if we zoom in on some of these biogeochemical cycles, like I said, into what we're really talking about, a fraction of a drop of seawater, we can see that some important processes at play that involve microbial interactions. So if we look over here, one of the microscale processes that has a big impact on carbon cycling in the ocean is the way that microbes interact with sinking organic particles, which we often call marine snow particles. So as you know, phytoplankton in the upper ocean fix carbon dioxide into living biomass during photosynthesis, but then many of these phytoplankton die or they're consumed by other planktonic organisms, and this leads to the formation of these marine snow particles, which are rich in organic carbon. And these particles, like you can see in this image of a sinking marine snow particle, are responsible for a massive flux of carbon from the upper ocean to the deep sea. About 10 gigatons of carbon every year are transported from the sunlit part of the ocean to the deep ocean, where some of this carbon is ultimately sequestered for up to hundreds or thousands of years. So obviously this has a big effect on the amount of carbon that can be exchanged between the upper ocean and the atmosphere. But microscale microbial processes have an influence on how much of this carbon is actually exported to the deep sea. So if we look at one of these marine snow particles under a microscope, after staining it with a nucleic acid stain, we'll often see something that looks like this, where all of these little blue dots are heterotrophic microbes, including bacteria and archaea, which heavily colonize these marine snow particles because of their rich source of organic carbon, which the microbes can consume. These microbes use extracellular enzymes to break these particles down and consume the carbon. And this can recycle this carbon before it reaches the deep ocean. And this, of course, has an influence on the ultimate amount of carbon that's exported. And like I said, this is all occurring within a microscale scenario. These particles are typically on the order of a fraction of a millimeter in diameter, and they're colonized by these bacteria, which influence this carbon flux. Another example of these microscale processes are in the top left here, and this is what I'm going to focus a lot of my talk on today. This is the interaction that occurs between phototrophic microbes, phytoplankton, and heterotrophic bacteria. And there's evidence that these interactions are highly important for both groups of organisms, and that this can ultimately have a big influence on governing the productivity of marine ecosystems, as well as chemical transformations at the base of the food web. And there's growing evidence that many of these interactions are highly specific and might take place within a microscale context where we see these cell-to-cell interactions occurring. An example of some of this is what we can see in this video. What we can see here is a seawater sample that we've taken, and you can see there are two organisms within this. There's a large organism with the spines, looks a bit like an insect. This is a diatom. It's a Chaetoceros cell, and you can see the little white dots buzzing around the cell, and these are heterotrophic bacteria. If you look closely, it looks like many of these bacteria are trying to maintain their position close to the diatom cell. And this, like I said before, is going to be a big focus of what I talk about in my talk today. So within my group, we're interested in these types of microscale processes, and we try to view the ocean from the perspective of a bacteria floating around in the seawater, rather than looking at it from a bulk scale. And from this perspective, what we think is like the ocean likely looks like to a bacteria is not a homogeneous soup of substrates and resources, but a structured environment where there's different microscale features which could each act as a niche full of resources for these microbes. And within these cartoon or images that we've come up with to describe this microbes view of the ocean, what I'm going to be focusing on today is one particular feature, and that's the interaction between phytoplankton and bacteria and how this occurs within this microscale scenario. So phytoplankton, as you know, fix carbon dioxide into living biomass, but a large proportion of this carbon that they fix ends up being released back into the seawater by the cells. Up to 50% or more of this carbon that they fix is released as dissolved organic carbon. So it's leaked into the surrounding environment, and within the region immediately surrounding each phytoplankton cell, within the area that we call the diffusion boundary layer, where the effects of turbulent mixing are minor, we expect to see chemical gradients emanating from the phytoplankton cell. And these are often rich in carbon. We know that marine bacteria obtain most of their carbon from phytoplankton in the environment. So this produces an environment or a microenvironment that bacteria might be able to take advantage of. And this area is being called the phycosphere, which is this area surrounding an individual phytoplankton cell, which is enriched in substrates, and it could be the interface for interactions between bacteria and phytoplankton. So everything I talk about today is going to be focused on this idea of the phycosphere and how bacteria and other microbes may be able to use behaviors to exploit this environment. And this is important because it leads to cycling of material released by the phytoplankton. As I said, it's a major source of resources for bacteria in the ocean. And more recently, we're starting to understand that this is probably the interface for really important symbiotic relationships that take place between phytoplankton and heterotrophic bacteria in the water column. So within this idea of these symbiotic or mutualistic relationships, we're now starting to learn a lot about the interactions between phytoplankton and bacteria. For a long time, it's been known that bacteria and phytoplankton probably benefit from each other. As I've mentioned already, phytoplankton release a lot of carbon into the water, which the bacteria use as a key growth source. And it had been thought that bacteria, through the remineralization of this organic matter, produce nutrients that the phytoplankton can then use. But in more recent years, we've got a much more sophisticated view on this, whereby we see that specific bacteria and phytoplankton are likely to take part in mutualistic associations, whereby really specific chemicals are exchanged between them, often in a reciprocal chemical exchange where they mutually benefit from these relationships. And there's been a lot of work in recent years in laboratory situations showing that certain bacteria and phytoplankton really have a strong interdependence on each other. Now, I'll talk more about some of these chemical exchanges later in my talk. So if we think about these close relationships between the phytoplankton and bacteria, if a certain bacteria really relies on a given phytoplankton for certain chemicals, then it makes sense that they probably should have close proximity to each other out within the vast expanses of the ocean where there's a milieu of different chemicals being mixed up. But this often isn't as simple as it might sound. So if we think about microbes out in the ocean, even though I mentioned they're highly abundant, when we look at their abundance and look at the distances between individual cells, these are often quite large. So for marine bacteria, the average distance, if we think of the bacteria being uniformly distributed in seawater, between two bacteria is typically around 100 cell lengths. So they're quite separated within the water column. And the distance between one of these bacteria and a phytoplankton cell, which they might be involved in one of these reciprocal chemical exchanges with, are also quite large. So if we think about a non-motile bacteria, something like a SAR11 cell floating around in the ocean, its chances of coming into contact with a phytoplankton cell are actually quite remote. It'll typically come into contact with a phytoplankton cell only once every 286 days. So obviously this isn't ideal if you want to be close to a particular phytoplankton partner. However, if we now consider that many marine bacteria are motile, so they can swim through the environment using helical flagella, which are these little tails that you can see on the bottom of this bacteria in the bottom right, which they use for propulsion to swim around through the environment. The encounter frequency between a phytoplankton and a bacteria increases dramatically. Now, one of these motile bacteria is likely to come into contact with a phytoplankton cell about 9 times every day. So you can see there's a big change just driven by this motility. If we include another behaviour related to motility on top of this, called chemotaxis, which is the capacity of bacteria to direct their behaviour according to chemical cues and chemical gradients in their environment, then this means that the bacteria are likely to come into contact with the phycosphere and be able to maintain their position within it much greater. So much so that we might expect to start to see bacteria clustering or maintaining their position close to a particular phytoplankton host. And that's what we think is happening in this image in this video that I showed earlier, where these bacteria are swimming close to this diatom cell. They're likely using chemotaxis to maintain their position close to this diatom, which is probably releasing organic carbon into the environment. Now, this idea has been around for quite a long time. It was first proposed in the early 1970s in this paper by Bell and Mitchell, where they showed that many marine bacteria exhibit chemotaxis towards the chemical products produced from phytoplankton. And they suggested that this bacterial chemotaxis probably helps a bacteria maintain itself close to this source of organic material, which is the phytoplankton. And they were the first people to come up with this term, the phycosphere, which is this region surrounding an individual phytoplankton cell. But since then, although a lot of people have presumed this to be important and to play a role in ecologically important interactions between phytoplankton and bacteria, there hasn't been a lot of real concrete evidence for the phycosphere and the role that behaviour might play in allowing bacteria to take advantage of it. So this is something that I've been interested in for several years now, and first started studying when I was doing my postdoc in Roman Stocker's lab at MIT about 12 to 15 years ago. And at that time, we wanted to come up with an approach to try to mimic this phycosphere within a controlled experimental scenario so that we could really track the way that bacteria behave within this microenvironment. So we used a technique called microfluidics, which involves the fabrication of small chips And on these chips, we can etch complex patterns and make things like microchannels within which we can control very carefully small volumes of fluid and create chemical gradients. And in this photo, you can see one of these microfluidic chips. To give you an idea of its size, that in the background, you can see that's the objective lens of a microscope. So this is a couple of centimeters long and a few millimeters wide. Now, we use these microfluidic channels to try, like I said, to simulate the phycosphere. And using one of these systems, what we did was injected in bands of phytoplankton-produced chemicals. So we'd take phytoplankton cultures, extract the chemicals that they exuded, and inject them into the channel. And that's what you can see here. This is a band of these phytoplankton exudates, which we're imaging after adding a fluorescent stain. And this is about 100 micrometers wide in diameter. And this diffuses across our microfluidic channel at the same kind of spatial and temporal scales you'd expect to see in a real phycosphere in the ocean. We then put marine bacteria into this channel and use video microscopy to track their behaviors. And that's what we can see here. These yellow lines are the swimming paths of individual bacteria within the channel, and they're swimming around. And you can start to see that they're not uniformly distributed across the channel. They're maintaining their position in the middle of the channel where we had this patch of phytoplankton exudates. So this is some of the first evidence that we had that bacteria could use this chemotactic behavior to hone in on these microscale patches and maintain their position within them, where they're going to be exposed to higher levels of the organic substrates released by the phytoplankton. We repeated these experiments across a wide range of different marine bacteria and to different phytoplankton exudates and found that this was quite a common feature amongst many marine bacteria. But there were a few shortcomings of this work, one being that it was restricted to the laboratory where we could perform it under a microscope. And this meant that we were restricted to using laboratory isolates of marine bacteria. So many of you probably already learned that from natural environments, it's often difficult to isolate bacteria from the environment. And in environments like the ocean, we can usually only cultivate a small fraction of all of the microbes that are actually in the environment. And that means in these experiments, we were using what you could describe as lab rats in terms of the microbes in these experiments. These were easily cultivatable bacteria which could grow up on nutrient-rich media. What it also meant is that many of these bacteria were highly motile and chemotactic. So these experiments could be giving us a biased view of the importance of this kind of behavior because we're using these lab rat type organisms. So our next step was to try to look at this in a more realistic framework. And the goal was to take this work back out into the ocean away from these laboratory experiments. And that meant that we had to develop a new experimental platform for trying to understand chemotaxis in the natural environment, whereby we could look at natural communities of marine bacteria that we didn't have to grow up or cultivate to start off with. And to do that, we developed a new piece of technology called the in situ chemotaxis assay, or the ISCA, which you can see in this image. Now, we used our same microfluidic fabrication approaches to make this system. And to give you an idea of its scale, this is about the size of a credit card. And you can see that it's an array of small chambers. Each one of these little circles is a chamber that has a volume of about 100 microliters. And it's enclosed from the external environment with the exception of these two small portals that you can see in each chamber. Now, we can use these portals to inject in chemicals, and that's what we use to inject in our chemical attractants or chemoattractants. And in the case I'm going to be talking about today, these are chemicals that are released by phytoplankton. So we can fill this ISCA device up with these chemicals, and then we physically deploy it into the seawater. So we put it into the ocean where it's exposed to natural microbiomes. And then what happens is the chemicals that are in these wells diffuse out of the well into the surrounding seawater, creating a chemical gradient close to the surface of this system, emanating out into the seawater where the microbes are. And if these microbes are motile and chemotactic, then they can potentially swim up these chemical gradients into the wells where the chemicals are coming from. So you could think of this as a microbial trap where we're using phytoplankton exudates as the bait, and the bacteria in the surrounding environment, if they like these chemicals, will swim in and they're captured within these wells. So what we're effectively trying to do is simulate these phycospheres within the marine environment in a way that we can capture the natural communities of bacteria. So we deploy this ISCA device in the environment for 30 minutes, then we retrieve it. We perform flow cytometry on the sample so that we can count how many bacteria swim into the wells and understand the levels of chemotaxis that have occurred. And then we perform DNA sequencing, and in this case, we use shotgun metagenomic sequencing to characterize the composition and potential function of the microbes that are responding to our different chemoattractants. So I'm going to talk about some experiments that we've done to try to test the hypothesis that different phycospheres or different phytoplankton will attract discrete groups of microbes that will end up in ecological relationships with those phytoplankton. So this is some work that was led by my postdoc, J.B. Rayner, who's worked in my lab for many years, and he was interested in this question of which bacteria swim towards which phytoplankton. So what he did was he grew up cultures of phytoplankton spanning all of the major groups of phytoplankton that we find in the ocean. He made them xenic, so removed all of the bacteria that were living in the cultures, and then used a solvent-based approach to extract the chemicals that these phytoplankton released into the media. So these are the chemicals that would be exuded into the phycosphere. And he put these in our ISCA and we deployed it in the environment. We did this in a number of environments. The experiment I'm going to talk about today is a deployment that we performed in coastal seawater off of the coast of Sydney, which is where we live. And we tracked the response of bacteria in the seawater to a suite of different phytoplankton exudates that you can see across the x-axis in this plot. Now, the way we quantify the strength of chemotaxis, as I mentioned, we perform flow cytometry, and we quantify chemotaxis using this chemotactic index, which is simply the numbers of cells that swim into our different chambers normalized to our control, which in this case is filtered seawater. So the reason we do this is because bacteria, just through random motility, will potentially end up in these wells. And so this is what we see within our filtered seawater, random motility leading to a small number of cells. But when we see a significantly higher number of bacteria within our treatments relative to this control, it indicates chemotaxis is occurring. And that's what we see. We've performed these experiments in a range of different places and over different seasons, and we find repeatedly that natural communities of marine bacteria exhibit chemotaxis towards a wide range of different phytoplankton exudates, like you can see in this plot. We see positive chemotaxis in a number of these cases. It varies depending upon the phytoplankton exudate, but we see that we can answer this first question, that it's not just in the laboratory where we see this behavior. Natural communities of marine bacteria can do this within the environment. So the next step, the next question is, if these communities are doing this, who amongst the communities are performing chemotaxis and towards what attractants? And this is where we start to use our metagenomic data, where we can characterize the taxonomy of the microbial community. In a moment, I'm going to show you a heat map showing you the diversity of the responding bacteria that have swum into these different chambers, which contain the phytoplankton exudates. So on this case, along the y-axis here, you can see color-coded are our different treatments, which are the phytoplankton exudates taken from different cultures. We have 5 replicates, and you can see that we get good coherence across our replicates. And when we look at the microbial community, with each one of these cells within the heat map indicative of a different prokaryotic taxa, we have bacteria as well as archaea that responded to our chemoattractants. There are 2 things that you can see. One is that across the replicates for each treatment, we see good coherence generally, and you then also see between our different treatments, there's different patterns. So different parts of the community are responding to different phytoplankton exudates. We don't just see one group of bacteria that are responding to all of the phytoplankton. We're seeing discrete populations responding to different phycospheres. We can look at this in another way. Here we can see where we've used a network analysis approach to show where certain bacteria end up within our different treatments. So to walk you through this, in this network, the large circles with the labels inside them are our different treatments. So they're our phytoplankton exudates. The small circles are different bacteria and archaea that we find in our ISCA wells. The lines between them, or the edges between them, are indicative of finding one of these bacteria or archaea within one of these treatments. Now, as you can see, there are a lot of different taxa here, and there are two different modes of connection to these treatments. In the middle, you can see there's a lot of bacteria that have multiple lines to multiple phytoplankton exudates. On the outside, you can see that there are some that are only attached to a single exudate. So these are what we call our generalist and specialist responses. The generalists are the organisms that are responding to a wide range of different phytoplankton. And in this changed version of the plot, you can see these are the coloured organisms. The different colours correspond to how many different treatments the bacteria were found in. So you can see there's one in the middle that was found in all 10 of the treatments. And then we can see the grey circles on the outside are the specialists, which are only found in one or two of the treatments. If we look at the distribution of these two different modes of association, we find that most of the bacteria that we found in these treatments fell in the specialist end of the regime. You can see most here on this plot, you can see our numbers correspond to the numbers of treatments the bacteria were found in, and you can see that most were found in 1, 2, or 3 treatments. Few were found in 7, 8, or 9. And so this indicates that the majority of the bacteria we found in these results were in the specialist category. So this answers one of these other questions: do we find specific microbial assemblages associating with specific phytoplankton? And it gives us this impression that within the seawater, we might have discrete phycosphere made up of different microbiomes with specific bacteria within them. So I've now answered a couple of questions. Can marine bacteria perform chemotaxis in the environment towards phycospheres? And do different communities respond to different phytoplankton? The next thing we were able to do is leverage our metagenomic data to try to look at what some of the repercussions of these responses are by looking at the functional profiles in these communities. So I'm sure that you've learned that metagenomics allows us to look at the predicted functional capacity of bacteria within samples. And so that's what we've done here. Now, the first set of functions that we looked at might seem obvious to you, and we did this purely as a sanity check. We look for genes involved in chemotaxis. So, of course, we'd expect to see that within our chemotactic experiment, within the wells, the bacteria would have heightened capacity for chemotaxis. And so we looked for genes involved in the chemotaxis pathway. And what we can see in this first column here, this is the bulk seawater outside of our experiment. And then when we compare it to what we see within our ISCA wells, and you can see in this heat map, the heat corresponds to the relative abundance of these genes. You can see there's an enrichment in genes involved in chemotaxis within our chambers. Like I said, this is not surprising at all. We'd be worried if we did not see this, but it was a sanity check to see that this approach was going to provide us with any interesting information. So the next thing we were interested in looking at was whether some of these bacteria that swim into these phycospheres that we've made might have the capacity to establish mutualistic partnerships with the phytoplankton. So I showed an image earlier which showed the many potential exchanges between phytoplankton and bacteria. One of them was this exchange here, which has come from some work in a number of laboratories where they looked at interactions between phytoplankton and bacteria and shown that some bacteria which benefit from the production of organic material from the phytoplankton can in turn provide benefits to the phytoplankton by producing what are called siderophores, which are specialized chemicals which are involved in binding of iron. Iron, many of you will probably know, is a limiting resource for a lot of phytoplankton. And it's been shown that bacteria that are enriched in these siderophores help the phytoplankton assimilate iron. And this can lead to an enhancements of phytoplankton growth. And it's been proposed that mutualistic partnerships between phytoplankton and bacteria are going to benefit from bacteria which have this capacity or have these siderophores. So we looked into the metagenomic data to see whether the bacteria that were swimming into our chambers showed this capacity. Again, I'm comparing it to what we see in the external seawater, and here we're looking at genes involved in the biosynthesis of siderophores by the bacteria. And we again found the statistically significant over-enrichment of these genes within our different chambers. So this indicates that these bacteria that are swimming into these phycospheres have this capacity to help the phytoplankton by producing these siderophores. Another example of many examples, I only have time to show you two today, but we saw many of these examples where bacteria had the capacity to help phytoplankton is in the provision of vitamins. So some of you might be aware that phytoplankton require B vitamins like vitamin B12, but many of them can't produce them themselves, so they rely on bacteria to provide these vitamins to the phytoplankton. And again, this has been shown to underpin interactions between phytoplankton and bacteria, whereby the bacteria benefit from a range of different organic substrates produced by the phytoplankton, and in return, the bacteria provide vitamin B12. So we look for genes involved in the biosynthesis and transport of vitamin B12. Same story as we saw before, we see a significant enrichment in these genes. So what we've seen across a number of these different pathways is that the phytoplankton, the bacteria that swim into these specialized phytoplankton-enriched microenvironments have a number of different functional capacities that will benefit a phytoplankton partner. And you could come up with the hypothesis that phytoplankton are releasing chemicals that actively attract bacteria that are going to bring these benefits towards them into their phycosphere to establish these interactions. So now for the last few minutes, going to move away from these these field-based experiments with the ISCA to target some more of these potential interactions that we've looked at within a laboratory scenario. One of these is some work by one of my PhD students, Neen Laroon, who was interested in the interaction between the marine diatom Actinocyclus curvatulus and its microbiome. She did a range of experiments, including looking at bacteria that she'd isolated from this culture capacity to perform chemotaxis towards the diatom. And she found that among these bacteria, one in particular, Ultramonas mediterraneae, showed a strong chemotactic response to the chemicals exuded by this diatom. She performed this using the same ISCA device I've talked about before, but in a laboratory scenario. She also performed metabolomic analysis of the chemicals released by this diatom and used some of these chemicals within the ISCA and showed again that this bacteria swam towards many of these. So within the system, we know that this bacteria can potentially swim towards the phycosphere of this diatom. She then performed some co-growth experiments after making the diatom exenic and showed that there's a mutualistic benefit from the association between these two. The bacteria was able to grow on the carbon produced by the diatom. We see a significant increase in bacterial growth when it's grown in co-culture with the Actinocyclus. And similarly, when we grow the diatom with the bacteria, we see an enhancement in growth of the diatom relative to an exenic situation. So there's this mutualistic partnership, which is underpinned initially by this chemotactic migration of the bacteria close to the diatom. We wanted to understand more about this, so another PhD student in my lab, Abiha Khalil, using the same model system, started to investigate what chemical exchanges might be involved in this relationship. And she was particularly interested in one group of chemicals called plant growth promoting hormones, which there's emerging evidence that many bacteria can produce. We know in terrestrial systems that bacteria that are associated with plants can provide these hormones to benefit the plant. And there's evidence that this is happening in phytoplankton bacteria interactions as well. So Abiha showed that this Ultramonas mediterraneae produces a large suite of different plant growth promoting hormones, often in quite high concentrations, which is surprising. We didn't expect a single bacteria to produce so many hormones. She then showed, using a technique where she knocked out the gene involved in the biosynthesis of one of these hormones called indole acetic acid, or IAA, using a CRISPR-Cas9 approach to knock out a gene involved in its synthesis. So she was able to make a mutant of this bacteria which didn't produce this plant growth promoting hormone, and then perform co-growth experiments. And she was able to show that by eliminating the capacity of this bacteria to produce these plant growth promoting hormones, it eliminated the promotion effect that this bacteria had. So we think that there's good evidence that this Ultramonas mediterraneae is benefiting this diatom by producing these plant growth promoting hormones after swimming into the phycosphere. And for the final couple of minutes, I'm going to talk about one final model system that we've looked at within our laboratory. This is work by another PhD student, Marco Giardina. He was helped by our postdoc, JB Renner. Here we were looking at the interactions between a picocyanobacteria, Synechococcus, which is one of the most important phototrophs across the ocean, and a heterotrophic bacteria, Marinobacter adherens. And here we've used a slightly different approach to what I've talked about up until now. Rather than looking at the growth promotion, we've looked more directly at the chemical exchanges between these two organisms. And the way we've done this is by using a technique called nanosims, which leverages imaging technology that allows us to look at the transfer of chemicals when we use stable isotope incubations of our organisms. So what we did in this experiment was we grew our Synechococcus, the picocyanobacteria, in a media which included 15N as the source of nitrogen. So 15N is the stable isotope of nitrogen. So we grew this Synechococcus within this stable isotope form of nitrogen, which meant we could then visualize this using our nanosims approach. We then co-grew the Synechococcus and the Marinobacter together, and we found that the Marinobacter was able to assimilate this 15N, which was released from the Synechococcus, likely in forms of dissolved organic nitrogen. So the Marinobacter benefited from this release of nitrogen from the Synechococcus. We then went back to our idea of whether behaviour is involved in this interaction. And here we took advantage of the fact that we had some mutants for this Marinobacter as well, including a chemotactic deficient and a motility deficient mutant. So we had a mutant where the gene Ki was knocked out. So this was a bacteria which didn't have the capacity to perform chemotaxis, and another one where the Fly-C gene was knocked out, and this is the flagellar assembly gene, meaning that this bacteria was non-motile. We performed exactly the same experiment as we did with the wild type. And interestingly, what we found was that the level of enrichment that we saw in the Marinobacter was heightened in the wild type relative to the two mutants. So what this tells us is that this capacity of the bacteria to swim close to the phytoplankton is important in this uptake of the nutrients released by the phytoplankton. And the final part to this story was that this behaviour was only one part of this reciprocal exchange. We also grew our Marinobacter up in a stable isotope. In this case, it was grown up in a media containing 13C, which is a stable isotope of carbon. And we're able to show that this 13C was transferred from the Marinobacter to the Synechococcus. So we know that Synechococcus can take up some organic carbon from the environment. And here we're able to show that organic carbon released by another bacteria was assimilated by this cyanobacteria. But interestingly, what we also found was, like we saw in the case with the Marinobacter taking up the chemicals released by the Synechococcus, we saw an increased amount of carbon taken up by the Synechococcus when we used the wild type of the swimming Marinobacter. So this tells us that the capacity of this Marinobacter to swim close to the Synechococcus not only benefited itself in bringing in chemicals released by the Synechococcus, but it benefited the Synechococcus cell by maintaining position close by and leading to a reciprocal chemical exchange. So there are a few examples of this little story that we're interested in my group, in how behavioral interactions, the swimming and chemotaxis by bacteria, can bring together potentially mutualistic partners, in particular the phytoplankton and bacteria. So hopefully today I've convinced you that next time you look at seawater, it's not just a homogeneous soup where everything's uniformly mixed up. It's a structured environment where behaviors of some of these microbes can facilitate interactions between individual cells. And if we scale this up, it can influence things like the growth of whole phytoplankton communities and important chemical exchanges in the marine microbiome. So with that, obviously, all of this work has been done by people that have worked in my group and other people that we've collaborated with. So I thank all of these people and thank Rachel for the invitation to speak to you all, and thank you for your attention.